Chapter 9, DNA-Based Information Technologies, Part 2. In this lecture, we will continue our discussion on DNA cloning techniques. Shown here is a schematic illustration of DNA cloning. In theory, DNA cloning can be divided into two steps. The first step is to generate a recombinant vector. And to generate a recombinant vector, one needs two things. The first one is a cloning vector, and the second one is a gene of interest. And in this case, it's a eukaryotic chromosome and a specific region of eukaryotic chromosome colored in red. First, the cloning vector is cleaved with a restriction endonuclease. Second, the DNA fragment of interest that is shown in red is obtained by cleaving this eukaryotic chromosome with the same restriction endonuclease used to cleave the cloning vector. Once the cleaved cloning vector and fragments of eukaryotic chromosomes are obtained, these fragments are then ligated together using a DNA ligase, and this results in the formation of a recombinant vector. In the second step of cloning, the recombinant vector generated is introduced into the host organism, and in this case, the host is E. coli or bacterium. Now, once this recombinant vector is introduced into the host, this vector gets propagated as long as bacterium multiplies. And this results in many copies of recombinant DNA containing the gene of interest inserted into the plasmid. Let us consider an example. Let us take a familiar vector, PBR322, and clone a foreign DNA in E. coli. Now, if you remember, this vector PBR322 has two selection markers, an ampicillin resistance and a tetracycline resistance. It also has various different restriction endonuclease sites. In the first step, PBR322 plasmids are cleaved by the restriction endonuclease PST1, which is present at the ampicillin resistance element. The same endonuclease is used to cleave foreign DNA of one's choice. And this results in these fragments, the cleaved fragments. Remember, when we use PST1, the ampicillin resistance is lost. The next step is to ligate these fragments together. And for this purpose, a DNA ligase is used. And once ligation is successful, the ampicillin resistant gene is disrupted, whereas the tetracycline gene remains intact. As you can see, all those successful ligation plasmids will have a red colored region flanked between the green colored region. This means that the gene of interest or the foreign DNA would be introduced in the place of the ampicillin resistance. The next step is to introduce this recombinant vector into the host organism, or E. coli in this case. And a technique called transformation is used. In the laboratory, small plasmids can be introduced into bacterial cells by a process called transformation. A classic transformation technique is electroporation. I'm not going to go in details as to how transformation is done, but in brief, what happens is that when electric charge is applied to bacterial cells that are competent, they can take up external DNA, and that is how recombinant plasmids get incorporated into the host organism. Once recombinant plasmid is introduced into the host organism, it is important to identify cells that contain this transformed plasmid. For this purpose, a technique called selection is used. And to select plasmids that are right, selection based on tetracycline is used, and bacteria is grown on agar plates containing tetracycline. And the rationale behind this is that not all plasmids that are transformed into the host organism are right, because all those that are shown by these red arrows contain foreign DNA that is flanked between the green regions, which happens to be the ampicillin resistant gene that is disrupted. Now, if the DNA ligase does not perform well, and if a plasmid did not get ligated properly, the ampicillin resistant gene does not get disrupted. For example, in this plasmid and this plasmid on the extreme right. Besides, not all plasmids that get incorporated by the foreign DNA have the right length. The one that is shown here has the ideal length of the foreign DNA that one needs to get incorporated into this plasmid. Whereas these two plasmids do not have the right length of the foreign DNA that is expected to be incorporated. For this purpose, an additional selection procedure is performed. And for this, the colonies that are present in the agar plates from the first selection process is repurified into additional plates. So the individual colonies are transferred to matching positions on additional plates. One plate contains tetracycline and the other tetracycline and ampicillin. All those host organisms or bacterial cells harboring plasmids that did not get ligated properly would grow in ampicillin plus tetracycline, whereas only those that have this specific gene incorporated at the right position by disrupting the ampicillin gene would grow in a tetracycline plate. After doing this experiment, this specific tetracycline plate can be further repurified, or the recombinant plasmid can be removed from bacterial cells and it can be sequenced. Researchers sometimes want to clone much longer DNA segments than typically be incorporated into standard plasmid cloning vectors, such as the ones that we saw before, like PBR322. Now, in this case, to meet the need, plasmid vectors have been developed with special features, as shown here. One such large segments of cloned DNA have been added, and this is a typical large DNA, about 100,000 to 300,000 base pairs. Now, to incorporate such a long DNA, plasmids like bacterial artificial chromosomes have been developed. A back vector is relatively simple plasmid, generally not much larger than other plasmid vectors. So to accommodate very long segments of cloned DNA, back vectors have stable origin of replication, that is shown here. Now, one of the uses of this origin of replication is to maintain this plasmid at 1 or 2 copies per cell. The low number of copies is useful in cloning large segments of DNA, like the one shown here, because it limits the opportunities for unwanted recombination reactions that can unpredictably alter large cloned DNAs over time. The back vector also include selectable markers such as CAM, which is an antibiotic resistant gene, and CAM stands for chloramphenicol. And chloramphenicol is an antibiotic, and CAMR is a chloramphenicol resistance gene. In addition, a LAX-Z gene required for the production of the enzyme beta-galactosidase is situated in the cloning region as shown here. Now, once a restriction endonuclease cleaves this LAG-Z region, the LAG-Z gene is inactivated. A DNA ligase then ligates the gene of interest into this specific endonuclease site. And if you observe this closely, the LAG-Z gene is now disrupted, so no beta-galactosidase is produced. And shown here is the recombinant back vector. Now, once this back vector is obtained, it can be electroporated into the host cell and can be selected for chloramphenicol containing cells, which means that if the plasmid is properly ligated, it will have a chloramphenicol resistance as shown here in this blue color. And only those cells with the recombinant back will grow. In addition, the second selection marker can be to look for XGAL. This means that if the vector is not properly cloned or not properly ligated, LAG-Z would not be disrupted. And all those cells without LAG-Z disruption or with XGAL expression are shown in blue. So, colonies with active beta-galactosidase and hence no DNA insert in back vector, they turn blue, whereas those that do not have beta-galactosidase activity are colorless, and those are the ones that will have the recombinant back plasmid. Another technique involved in DNA cloning is called as polymerase chain reaction, or PCR. Polymerase chain reaction is used to amplify DNA in the test tube. It can amplify regions of interest within linear DNA or can amplify complete circular plasmids. The PCR procedure relies on enzymes called DNA polymerases. DNA polymerases synthesize DNA strands from deoxyribonucleotides. For example, DGTP. DNA polymerases uses DNA template. Shown here is a template strand, and it's a single strand. DNA polymerases do not synthesize DNA de novo, but instead use pre-existing strands referred to as primers. So it uses a template and polymerases a primer strand based on the template by using deoxyribonucleotides. deoxyribonucleotides. And this is the principle of polymerase chain reaction catalyzed by DNA polymerases. The PCR procedure has an elegant simplicity. Basic PCR requires 4 components. First, a DNA sample containing the segment to be amplified. Second, set of synthetic primers or oligonucleotides that are complementary to target. Third, pool of nucleotides, in this case deoxynucleotides, because we are amplifying a DNA. Last but not least, a thermostable DNA polymerase, an enzyme that can catalyze the polymerase chain reaction. PCR involves 3 steps. In the first step, all these components are placed into a thermocycler. This is an instrument that has the capacity to heat the samples and simultaneously cool them. In the first step, the samples are melted at about 95 degrees centigrade, and specifically at this temperature it only melts DNA, which means that the double strand of a DNA becomes single-stranded. This is the reason why we are using a thermostable DNA polymerase, because the protein does not denature at 95 degrees C. In the second step, the samples are cooled so that the separated DNA strands can be brought together with the primers. This means that the primers that are added in the mixture bind to separate DNA strands at the temperature of about 50 to 60 degrees C. In the third step, the DNA polymerase uses the DNA as a template and adds deoxynucleotides onto the primers, and this is called as an extension reaction. So the polymerase specifically extends primers in the 5' to 3' direction, and after a round of elongation is done, these steps are repeated. Here is a schematic illustration describing general steps of PCR. Shown here in dark blue is a region of the target DNA that requires to be amplified. When you initiate PCR reaction, in the first step, when the specific DNA gets heated up, it denatures and becomes separate strands. In the second step, when the temperature is brought down to 50 to 60 degrees centigrade, the oligonucleotide primers anneal to separate strands, and they anneal to the complementary strand. For example, if the strand is 3' to 5', the primer, which is 5' to 3', anneals to that. And another primer that is 3' to 5' anneals to a strand that is from 5' to 3'. So basically, complementary sections of DNA gets annealed to each other. In the next step, the thermostable DNA polymerase uses the DNA strand as a template and adds deoxyribonucleotides to the primer. So it adds deoxyribonucleotides starting from here, and it extends this specific strand. So as you can see, a newly formed strand anneals with the 3' to 5' region of the original target DNA. The same way, the DNA polymerase continues polymerization reaction of the primer from 5' to 3', that is annealed to the 5' to 3' region of the original DNA strand. Now, this is just one step. When you repeat the same step, these oligonucleotide primers now uses the newly formed strand as a template because they anneal to them, and the DNA polymerase polymerizes subsequent strands. And that results in the formation of further newly formed DNA strands. After about 25 cycles, the DNA has been amplified about 10 to the 6-fold. So this is an exponential process. And after about 20 or 25 cycles, you can see that the region that needs to be amplified amplifies to an extent where DNA polymerase no longer needs to amplify. And this is the basic step of PCR. DNA electrophoresis is an invaluable technique that is used during DNA cloning. Negatively charged DNA migrates to the anode in the presence of an electric field. Agarose, that is the matrix of the gel, hinders the mobility of DNA molecules. Mobility of DNA depends on the size and the shape. Small and compact molecules move faster. The practical uses of DNA gel electrophoresis are plenty, from DNA analysis to DNA purification to DNA protein interaction studies. And in case of DNA cloning, DNA gel electrophoresis is used to purify the cloned plasmid. PCR has many applications. Besides, there are many other techniques that are adaptations of PCR as well. What are the applications of PCR? Cloning. By using flanking primers to replicate and amplify a specific segment of DNA. Side-directed mutagenesis, or in this case, oligonucleotide-directed method. We haven't talked about side-directed mutagenesis. I will be explaining as to what side-directed mutagenesis is in the upcoming slides. In this method, specifically, one uses sandwich primers to make small changes to the segment of DNA and then amplify the altered DNA. Briefly, side-directed mutagenesis technique is used if one wants to mutate an amino acid in a protein of one's choice. And the way one does that is by mutating a codon in the DNA that translates the protein. DNA fingerprinting. This has immense potential and application as well. PCR techniques are used here, and primers are used for specific chromosomal locations to amplify and determine number of short tandem repeats for purposes of identification. I'll be talking about DNA fingerprinting later on. What are the adaptations of PCR? There are other techniques that have evolved from PCR, and one of them is reverse transcriptase PCR or RTPCR. Specifically, this technique is used to convert RNA to DNA and amplify specific segments. In many cases, DNA sequence may not be available. In such circumstances, an RNA needs to be converted to a DNA, and for this purpose, reverse transcriptase PCR is used. Quantitative PCR or qPCR is used to show quantitative differences in gene levels, and this is a very powerful technique as well. We will talk about these techniques later on. Let us try and understand as to what side-directed mutagenesis is. In side-directed mutagenesis, one alters the genetic code. And there are 4 different points, and it is important that you understand these points. First, understanding the function of proteins often requires that a specific amino acid residue be mutated. What does this mean? Let us imagine that if you are studying the function of a protein, let us say it's an enzyme. Now, to study and characterize the function of an enzyme, it is important to understand what individual amino acids in the active site really contribute. To understand that, one typically mutates a specific amino acid in the active site or in the region of interest in the protein of their choice. Now, how do one go about mutating an amino acid? And that brings us to the second point. To mutate an amino acid, one typically change the nucleotide or nucleotides in the coding DNA and express the mutated gene. That is how proteins are mutated. If you remember the central dogma of molecular biology, proteins are evolved from DNA, right? Transcription leads to translation. And that's what brings us to the central dogma. Now, another important point is that an amino acid is represented by 3 bases called as codons. And we'll talk about this in detail in chapter 27. The third point is that side-directed mutagenesis usually relies on chemically synthesized mutated primers that are incorporated into newly synthesized DNA. So side-directed mutagenesis is very similar to PCR. One of the main differences is that the primers that are used are slightly different as compared to a normal gene amplification or a DNA amplification. And we'll talk about that in a moment. But the point remains that the primers are chemically synthesized. Once you mutate a plasmid or a gene of your interest in a plasmid, you confirm the mutation by sequencing the plasmid. And this is important because sequencing your plasmid is the only way to confirm that a specific base or a group of bases are mutated. Side-directed mutagenesis can be performed using 2 different approaches. In the first approach, a recombinant vector harboring a target gene can be cleaved with a restriction endonuclease of one's choice, and a mutation containing gene can be inserted back into the cleaved recombinant vector using a DNA ligase to obtain a new recombinant vector with a mutated gene. In the second approach, a pair of synthetic and complementary oligonucleotides with a specific sequence change at one position can be hybridized to a circular plasmid with a cloned copy of the gene to be altered. The mutated oligonucleotides that is shown here acts as primers for the synthesis of full-length duplex DNA copies of the plasmid that contain the specified sequence change. Now, when PCR is performed, full-length plasmid is obtained, as opposed to a linear target gene of one's choice, as seen in PCR experiments before. Now, these plasmid copies are then used to transform into cells. And once these are transformed, they need to be sequenced. Now shown here is a representative example for a protein named REC-A. Wild-type REC-A, or the normally occurring REC-A protein, has a representative sequence. And the reason why this region of the DNA is shown is because this codon AAA is mutated in the mutant REC-A. And in this case, a lysine is mutated to an arginine. And the codon that represents lysine is AAA, whereas the codon that represents arginine is CGC. So, in mutating wild type to a mutant, one can actually sequence and obtain the sequence results from an automated sequencer. And this is showing the mutated sequences in the bottom. And as you can see, CGC is clearly shown here as compared to AAA in this case. So this technique, or side-directed mutagenesis, is extremely powerful, and it's utilized by biochemists to mutate one or multiple amino acids in protein of interest. Many additional adaptations of PCR have increased its utility. RTPCR, or reverse transcriptase PCR, is one such adaptation. RTPCR involves amplifying RNA instead of DNA. Here, reverse transcriptase is used instead of a DNA polymerase. In case of RT-PCR, an RNA strand is usually amplified in the beginning. The technique is very similar to PCR in the sense that when an RNA strand is amplified, primers are added and ribonucleotides are included in the reaction mixture. Then reverse transcriptase is added, which then transcribes or converts the RNA to DNA. Once DNA strand is made from the RNA template that was originally present, the remaining cycles can be carried out with DNA polymerase, and normal PCR can be performed. So this technique is an advantage in the sense that RNA can be used to obtain the information about the gene. RT-PCR can be used to detect sequences derived from living cells as opposed to dead tissues. So in living cells, DNA is being constantly transcribed to RNA. So this RNA can be taken and converted back to DNA by using RT-PCR. Another adaptation of PCR is called qPCR, or quantitative PCR. qPCR, or real-time PCR, is used to estimate the relative copy number of particular sequences in a sample. For example, qPCR can quantitate the amplification of specific genes in diseases like cancers. How is qPCR performed? Well, qPCR is performed typically using a primer that is complementary to the sequence of interest. And if you consider the primer has a fluorophore that is attached to one end, which is quenched by a quenching molecule. Now, the amount of PCR product present is determined by measuring the level of a fluorescent probe, which is this, attached to a reported oligonucleotide, that is this, which is complementary to the target DNA, that is to be amplified. The probe fluorescence is initially not detected due to this quencher, and that quenches the fluorescence of this fluorophore when they are present close to each other. When the reporter oligonucleotide pairs with the target DNA that is complementary to this oligonucleotide, in a copy of amplified DNA segment, the fluorophore is separated from the quenching molecule, which is here, and as a result, fluorescence increases. And the result from such an experiment usually looks like this. And this is a graph of signal on y-axis versus PCR cycle number on the x-axis. As the PCR reaction proceeds, the amount of the targeted DNA segment increases exponentially, as you can see. And the fluorescence signal also increases exponentially as the oligonucleotide probe anneals to the amplified segments. Now, after many PCR cycles, which is given by PCR cycle number here, the signal reaches a plateau, and that is shown here. All right, the signal goes up, increases exponentially, and reaches a plateau. Now, this reaches a plateau as one or more reaction components become exhausted. So, when a segment is present in greater number than another, its amplification reaches a defined threshold level earlier. For example, sample 1 reaches plateau much earlier than sample 3. This means that the level of sample 1 reaches the threshold earlier and has a higher amount of the sample present in the reaction mixture. Whereas no template that is shown here, which has a very small slope, it increases very slowly in this case, and it has a background signal, and it does not have any DNA that is complementary to the oligonucleotide primer with a fluorophore. And so that's a control. And this is how a qPCR experiment is performed. DNA cloning is also performed to express proteins of one's choice. And one of the reasons is because one wants to study the protein product. Special plasmids called expression vectors contain sequences that allow transcription of the inserted gene. These expression vectors are slightly different from cloning vectors that we saw before. Some major differences are as follows. Expression vectors have something called as a promoter sequence and an operator sequence, and these are bacterial promoter or operator sequences. Now, depending on which organism one is intending to express their proteins, one must choose expression vector depending on the organism. Now, in this case, this specific expression vector is for a bacterial expression, and hence a bacterial promoter and operator is usually embedded in this expression vector. There is a code for ribosome binding site as well. Usually, the gene of interest is cloned after the ribosome binding site. In addition, there is a transcription termination sequence as well. Once the gene of interest is cloned in this vector and inserted into expression system of one's choice, proteins are usually expressed and amplified. Many different systems are used to express recombinant proteins. Bacteria, especially E. coli, remain the most common host for protein expression. The reason is because E. coli is versatile and the procedure is relatively inexpensive. One of the major limitations is that when a eukaryotic protein is expressed in bacteria, some of these proteins can be intrinsically disordered when expressed in bacteria. And one may not be able to express and purify a fully folded eukaryotic proteins in bacteria. To circumvent this issue, scientists use yeast system to express and purify eukaryotic proteins. Yeast system is also relatively inexpensive. However, the limitations are pretty similar to bacteria, and certain eukaryotic proteins cannot fully fold in yeast expression systems. Insect and insect virus systems are used extensively to express and purify eukaryotic proteins that are difficult to express otherwise. The limitations involve labor-intensive procedures, but expressed proteins are physiologically relevant with proper post-translational modifications in them. Mammalian cell culture is another technique that is used, but it is seldom used to express and purify proteins. Mammalian cell culture is often used to validate a specific protein and its binding partners. One of the reasons is because this technique is relatively expensive. After recombinant proteins are expressed, they need to be purified as well. Purification of natural proteins is difficult. Recombinant proteins can be tagged for purification, and the way one can do this is by genetically modifying when performing cloning. So what is a tag? The tag that is attached to a protein binds to an affinity resin, binding the protein of interest along with it to a purification column. I'll explain this principle in the next slide, but let us see as to what constitutes a tag. A tag can be a protein or a peptide. This table shows you various different tag proteins or peptides that are commonly used. When protein A is used, the Fc portion of this antibody is used as an immobilized ligand in the column. When polyhistidine is used, nickel beads are used as immobilized ligand in a column for purification. One of the reasons is because histidine has a very high affinity towards nickel, and it forms coordinate bonds with nickels and can get stuck in the column, thereby also tagging along the protein that it is attached to. Another example is glutathione S-transferase. It's a protein which has high affinity towards glutathione. Let us consider an example. This example involves purifying a recombinant protein attached to a GST tag. The gene for GST tag is fused to the carboxy terminus of the gene for target protein, thereby making a fusion tag in the cloned plasmid. Once the plasmid is cloned, it is expressed in an expression system of one's choice. The tag protein is expressed in the cell and is present in the crude extract when cells are lysed. The extract is then subjected to affinity chromatography through a matrix with immobilized glutathione. As I said before, GST or glutathione S-transferase is a small enzyme that binds glutathione, which is a dipeptide. Now, this glutathione is immobilized in this specific column. And the matrix is made up of immobilized glutathione. So when the GST tagged protein is passed through this column, only the GST tagged proteins bind to the glutathione in the matrix, thereby retarding its migration through the column, while all the other proteins are washed through rapidly. The tagged protein is subsequently eluted with a solution containing elevated salt concentration or free glutathione. So if you add free glutathione through this, glutathione then replaces all the protein that is bound here, and whatever protein that is bound to the matrix elutes. And the fusion proteins can be eluted using this procedure. This is a classic example of how recombinant proteins are purified using specific tags.